This video will be an overview of the genetics material required for a GOB class in chemistry, an introductory chemistry class. Okay, so there's some vocab that we should definitely be aware of. Chromosomes and genes, you've probably heard some of this vocab in popular media. Maybe you've looked at one of those popular sites that can tell you about your genetic information. So let's have a look, and it's definitely important vocab for any assessment. So select the pen, chromosome. I would like my students to know what a chromosome is. So it's essentially the storage vesicle that DNA is kept within. So let's say stores DNA. It's how DNA is packaged. Humans. Now, this next bullet point is a loaded question. There are many diseases where you can have more than 23 pairs, but let's say that the intentional design for a healthy human is to have 46 chromosomes, so 23 pairs. There's a popular DNA company called 23andMe, and it's based on this idea that humans are intended to have 23 pairs of chromosomes. So we have 46, which is just twice 23 individual chromosomes. And again, each of those chromosomes stores specific genetic information. A gene is the prefix for genetics. So this is a word that most people have heard of. So the study of genes is genetics, and it's a portion of the DNA molecule responsible for different proteins. So not all not 100% genes. You know, there are some parts of DNA that has no genetic function. So DNA and genes are not the same thing. Genes are the I guess I have to be careful. I was going to say active part of DNA, but you know, the non-active part, by active I don't mean essential. You know, it's reasonably agreed upon that it's essential that the non-active parts of DNA play a role too. DNA itself, DNA is an acronym for, let's see, it's probably on the next slide, deoxyribo, it's all one word, nucleic acid. So D for deoxy, R for ribonucleic, and then A for acid. DNA molecules contain several million nucleotides. This is a structure we'll see in a second. A nucleotide is the proverbial brick of DNA. So the building block of DNA that we'll see in a second. There's another, not a building block of DNA, but a precursor to DNA. So let's say the ancestor of DNA called RNA. RNA is ribonucleic acid. So ribonucleic acid, ribonucleic acid RNA. And these are typically shorter chains. They don't have the secondary structure that DNA can have eventually when we look at secondary structure. So a primitive version from which there's evidence that DNA evolved. Again, we said that DNA is contained in the chromosomes. So imagine the chromosome is like, I don't know, not to totally lose any kind of standard, but it's like the suitcase in which DNA is packaged. For example, when a cell replicates, so that you can make sure that when you arrive at your new cell destination, you haven't forgotten your shorts or your flip-flops, etc. Okay, so we've hammered the chemistry out of that slide, so let's move on. So some vocab there. Okay, a lot more vocab here. Nucleic acids are unbranched polymers. Remember from our organic chemistry video, a polymer, poly just means many, and mer just means copy. So a polymer is a large molecule that has many repeating copies of a simple structure. So essentially, a polymer is a wall and the many copies of the bricks. The bricks here are the nucleotides. So again, the nucleic acid, this is the wall, the proverbial wall of the DNA, the deoxyribonucleic acid, and the nucleotide, remember, is the brick. It's the many copies of which the wall is based. Now we're going to look at the structure of the bricks in a couple of slides, but when we do, we'll see that the bricks or the nucleotides, in fact, I'll probably just come back to it. I think that's what I'll do. Rather than say, well, eventually we'll look at it, we'll just come back to it. So let's worry about pyrimidines and purine bases in a second. Again, we've mentioned that there are two types of nucleic acids. There's deoxyribonucleic acid. There's ribonucleic acid. Again, we're going to look at the structure of those in a second. There's a lot of consequences to the difference of those structures. We know, if from nothing else than from Jurassic World, I'd say Jurassic Park, but you'd have to be born in the 90s. Well, you'd have to be born in the 80s to know what Jurassic Park is all about. But if you're born a bit more recently, you probably know only Jurassic World. Then you probably know DNA stores genetic information. So, for example, if you want to make a T-Rex or a Velociraptor, I would think again, but let's say you wanted to anyway. You would need to replicate the genetic information. You would need the DNA. And mercifully, nobody has the DNA of a T-Rex. Otherwise, we'd all be eaten by one because there'd be one behind you. But the DNA stores genetic information. So I like to think that if you wanted to send a message in a bottle, you would write some information, put it in a bottle, throw it at sea, and it would eventually land upon a shore somewhere. DNA, I like to romanticize this as like a message through time. So it sends a proverbial message through time. You know, so 2 million years ago, or whenever the most recent argument says that humans became humans, you could say this is the message for a human, and 2 million years later the message is still being delivered. The message can be improved, but there's still a message through time. So that's how I like to think of DNA. And DNA and RNA can also send a message through time, but since the evolution of DNA essentially instructs RNA to do its bidding. So RNA does a lot of the heavy lifting, but DNA regulates the message. Okay, so a lot more vocab in that slide. Okay, this is a chemistry class, so let's look at some chemical structures. So we mentioned earlier, and here's where I need the earlier mention. We mentioned earlier that there were nitrogenous, that just means contains nitrogen. There were bases, purines, and pyrimidines. So these are bases, and this is how the message through time is transmitted. So the message through time is transmitted via pyrimidine and purine bases. So let's look at what those bases are. They're obviously very important. So we've got pyrimidine bases. Well, first of all, before we look at the bases are on this slide. Let's consider the brick of the DNA. So these are nucleotides, and we've said already that these are the bricks or the building blocks of our message in a bottle through time, or our DNA. Okay. So nucleotides are, they contain 3 components. Notice that they contain a phosphate group. So we'll call this our phosphate. Phosphate, the suffix H just means contains oxygen. Phosp obviously means phosphorus, so we have phosphorus here, and we've got lots of oxygens around it, so that's our phosphate group. We have the Hayworth projection of our ribose sugar. So this is a Hayworth projection of our sugar. And then we have our base over here, our nitrogenous base. So there's 3 components. Again, you can see here the phosphate, the Hayworth projection of our ribose sugar, and our nitrogenous base. And then again, the phosphate, the Hayworth projection of our ribose sugar, our nitrogenous base. Okay. Again, down here, our phosphate, Hayworth projection of our ribose sugar, nitrogenous base, and you won't be dazzled to hear that this has a phosphate group, Hayworth projection of our ribose sugar, nitrogenous base. Okay, so lots of information there. Let's clear up the slide a wee bit now that we've pointed all of that out. Let's tidy things up a bit. Okay. Couple of things that we should notice. First of all, what's the difference between a pyrimidine and a purine? Well, a pyrimidine has a single ring and a purine has a double ring. So you can see here these bases have a single ring. So these are all pyrimidines. These bases have a single ring. They are pyrimidines. We have a hexagonal nitrogenous base here with a single ring. So these are pyrimidines. These bases, on the other hand, have double rings, a hexagonal ring or a hexagon, and then a pentagon or a pentagonal ring. Double rings means these are purine rings. Okay, so we have pyrimidine rings and purine rings. Okay, let's clear that up now that we've pointed that out. Okay. Notice that we can probably see it more clearly. Yeah, it's coming up. We don't have to point it out here. It's coming up on another slide. Notice that if we focus on our Haworth projection of our ribose sugar, here, this 5-membered ring, this is the sugar part of DNA. Let's look at the 2' position here. So for my students at least, it's not essential that you know the numbering sequence of the ring atoms in the base, but it is essential that you know the numbering system of the ring components in the sugar. They all have primes and they're labeled clockwise. So 1', 2', 3', 4', 5'. So for my students at least, that is essential. Let's look at the 2' carbon. Only the 2' carbon, which is here. 2' carbon. So we're considering the 2' carbon. If there's an OH, we know the nucleotide is RNA. So these are from RNA because they have an OH on the 2' carbon. However, if there is no OH, then we know it's a DNA nucleotide. So we know that this is a building block for DNA because it's deoxygenated on the 2' carbon, and the absence of a D here in these abbreviations means that these are building blocks for RNA. Why might the 2' carbon be deoxygenated? Well, from a chemist's point of view, oxygen activates a carbon bond because oxygen is highly electronegative. And we'll see in the metabolism chapter that oxygen can be used chemically or can be interpreted chemically to activate a carbon bond or a carbon atom for bond cleavage and other such things. So we know that being deoxygenated on carbon 2, we know that that sugar is more stable. So that would be a consequence of the 2' carbon being deoxygenated. Why might that be useful? Well, might be good for preserving our message in a bottle. So hypothetically, that is why DNA is a better store of information than RNA, because it's more stable. Okay, so I think we've exhausted this slide, so moving on. Just in passing, there's some important nucleotides. Notice these, if we look at the 2' carbon of the Haworth projection of the ribose sugar, these are both from RNA. If they were DNA, they would be deoxygenated here. Okay, so most people have heard of ATP. This essentially is biological energy. And essentially, this is a waste product of ATP. Essentially, the only difference is if we lose this phosphate group from ATP, where T just means tri, so we've got 1, 2, 3. So this is triphosphate. We've lost the first one, so we only have 2 diphosphate. Okay, so it's an exhaust product. And we can see why ATP is an energy source. We've got these negative charges here. So we've got repulsion, electrostatic repulsion, due to electrostatic repulsion. So if you consider just basic Coulomb's law, like charges repel. So this becomes a phosphate gun, if you like, which fires off phosphates, which essentially are just sources of oxygen, to activate carbon compounds. And we'll see that later. So just in passing, we can see that these are nucleotides, very important nucleotides. We know that they're nucleotides because they have phosphate groups, ribose sugars, and bases. We know this is a double ring, so we know this is a purine type base. Again, we know this is a double ring, so it's a purine type base. So moving on. Let's look in a bit more detail at the 5 bases. I'm not really that concerned if my students know the exact name. What I would require is that you know the single letter abbreviation. Okay. So there is complementary base pairing. Well, first of all, these are the 2 templates. Let's not get ahead of ourselves. These are the 2 templates. We said the single-membered ring is the pyrimidine. The double-membered ring is the purine. So there are 3 types of pyrimidine: uracil, thymine, and cytosine, which are abbreviated U, T, and C respectively. And there are 2 purine bases, adenine and guanine, symbolized by A and G respectively. Notice that one of them is only found in RNA. So this is the odd one out. So this is the odd, almost like a relic. Uracil is only found in RNA. So we're going to see pretty soon what would be essential here is that we have complementary base pairing. So adenine is going to essentially bond, or hydrogen bond, specifically hydrogen bond, with thymine. And guanine is going to preferentially hydrogen bond with cytosine. Now this is in DNA. In RNA, we don't have thymine. So in RNA, thymine is replaced with uracil. You can see the similarity of the structures. There's a methyl group here. It's been demethylated here. So in RNA, we have adenine hydrogen bonding preferentially with uracil. And we have guanine preferentially hydrogen bonding with cytosine. So this is what I would highly recommend that anyone memorize, and certainly my students should memorize. Okay, let's look at a piece of the wall. So this is a piece of the wall where the wall is DNA. So first of all, we can see the bricks. We know that that's a nucleotide because it's got the phosphate group, it's got the Haworth projection of the sugar, it's got the base. We also can see that the 2' carbon of the ribose sugar is deoxygenated. And that's what tells us it's DNA and it's not RNA. If it were RNA, it would have an oxygen there. And in this cartoon here, notice, well, you can notice probably a bit clearer if I delete something. Notice we have the 5' end and the 3' end. The 5 and the 3' end. Let's clear this up. That's important. Let's see why is that important? It's important because of this statement here, which is conveniently placed. So it reads, it's kind of small, but it reads the DNA sequence is read from the 5' end to the 3' end in the direction given by the arrow. So let's clear this up a bit so we can actually see where the 5 and 3 end come from. Let's clear this up a little bit. Remember, I alerted my students earlier to the numbering of the ribose sugar component. So this is the 1' carbon, this is the 2' carbon, this is the 3' carbon, this is the 4' carbon, and this therefore is the 5' carbon. So now you know why it's called the 5' end. It's the 5' carbon end. Let's look at the 3' end. We've got the 1' carbon of ribose, the deoxygenated 2' carbon, 3' carbon, 4' carbon, 5' carbon. So again, notice that we have the exposed 3' carbon end here. Why not just call it the 5' end? Well, look at how each brick is laid. We are laying, if I get rid of this little label here, notice that we have a brick or a nucleotide connects from the 5' end to the 3' end. Between the 5 and the 3' end, we have these phosphate linkers. So these linkages are called phosphodiester linkages. Phosphodiester linkages. This is the bonding between nucleotides in DNA and RNA. So phosphodiester, well, phospho means it contains phosphorus. A diester means it's two esters. So a phosphoester would be a P double bond O with an O. A diester means you have a plane of symmetry to the other side as well. So if we draw a plane of symmetry right down here, we've got an ester that way. We have an ester that way. So we've got a diester. Di just means two. And phospho means it's phosphorus rather than carbon. So it's a phosphodiester. And it's a link between the nucleotide bricks. Okay, so that's an important, so essentially this is how we lay the foundation for DNA. Now, there's a time and a place for the weeds, and there's a time and a place to get in a helicopter, fly above the weeds, and realize that there's a beautiful forest there. Chemists need to be able to understand what all these atoms mean and what have you, but at some point we don't need all the detail. We need to focus on what's really important. And so for the most part, when DNA is studied, certainly at this level, we just look at the base pairing. So kind of analogous to the ribbon diagrams in our protein chapter, this is a double helix strand of DNA. You can see just about, it's kind of small, so maybe I'll zoom in a bit. You can see here that we have the 5' end, a complementary strand, the 3' end. So essentially we have, if I were to elongate this, we'd have the 5' end and the 3' end of one strand. And then alongside we'd have the 3' end of another strand and the 5' end of another strand. And there'd be hydrogen bonding interactions between them. And then you just coil into a helix. Okay, and that's what you have here. Notice that we have the complementary base pairing. So we have the guanine here. Let's get my red pen back. So we have the hydrogen bonding. Between guanine and cytosine, and between thymine and adenine. This is the complementary base pairing. Okay. So I can zoom out a little bit of that. Okay, so again, I definitely want to emphasize that you should memorize this complementary base pairing. Here's a cartoon that you'll typically see in an assessment just because it's easy to produce. Again, you always have the two strands of DNA in anti-parallel orientation, so the 5 to 3 end and the 3 to 5 end. And again, you've got this AT and G and C, and you tend not to have anything else. So I recommend you get comfortable with that. Let's have a look at this one. So again, we said we've got our AT base pairing. You can see the actual bases here, and we have our GC base pairing. So the fact that we have T means that this is DNA. So notice that we always have base pairing between a pyrimidine and a purine. So it's always pyrimidine to purine, pyrimidine to purine. It's never pyrimidine to pyrimidine or purine to purine. It's always one of each. And this should say nanometer. So that's a little typo there, nanometer. Okay, and we can see the hydrogen bonding here. So we've got an amine group here. Again, we have, let's see, an amine group here. We've got an amine group here, and amine group here. Let's see. Now, 1, 2, 3, 1, 2, 3. Okay. And we have, okay, so here's our hydrogen bond here. Here's our hydrogen bond, here's our hydrogen bond. I'll do it in black here. So we can see, not only can we see the hydrogen bonding, but we can see the almost regularity of the separation. So this uniformity stabilizes the structure, and it's this uniformity that that gives it preference over pyrimidine-pyrimidine base pairing or purine-purine base pairing. So again, to emphasize that point, this is what is observed in DNA and RNA. We don't observe two pyrimidines, we don't observe two purines, we do observe one of each. And it's that uniformity. If we were to have a mixture of all three, you'd have, so all three would give you a variation in base separation. Whereas only one gives you uniform base separation. And this can lead to stability of the structure. Okay, some vocab here. Incidentally, we're about halfway through the slides. There's about 19 slides here. So we're about halfway through. Try and keep it under an hour. So some vocab. So what does DNA do? Well, DNA is like a factory, and factories have to make more factories. But then at some point, the factory has to make a product. So the product that's made from the DNA factory is protein. The message in the bottle is the instructions to make protein. But the factory has to make the protein factory has to make more factories. So we have to replicate the DNA just to make more copies of it. And then we have to start making product. So we start making product via transcription. We transcribe the message from the DNA and we relay the message to RNA molecules, and then the RNA can then go on to help manufacture the protein. So replication, transcription, translation, these are key vocab words that we're going to start to look at now. This next slide has a nice way to look at it. So we've got this fork in the road here. So let's say this is the factory, to use an analogy. This is our factory, and our factory makes product. So this is the product of our factory. Well, we need a lot of factories. We want to make a lot of product. So to make enough product, we need enough factories. So we have to replicate and make more factories. So DNA has to be able to replicate itself, and then once there's enough of it, it has to be able to start the transcription phase to RNA and essentially translate that to protein. Notice that this could happen anyway. So this is the ancestral path to protein. This part here is a later evolution. Okay, here's a diagram of what this might look like. So you've got your coiled strand of DNA here, coiled helix of DNA here. Here it starts to be uncoiled, ready for either replication or transcription. And notice that we have, first of all, I want to point out we have a leading strand of DNA and we have a lagging strand. Okay, we've got our bases here, that's fine. I'm not really interested in the bases in this slide. We can zoom a little bit though. Okay, so let's look at why I've called it leading and lagging strand. Essentially, first of all, we're going to read in the 3 to 5 direction. Okay, and we're going to synthesize in the 5 to 3 direction. So here's the original strand here called old, and it starts off 3 here. So we start to make the new strand here in the 5 to 3 direction. So 5 to 3 is the new, because we're reading in the 3 to 5 direction. How do I know what to make? Complementary base pairing. So we synthesize in the 5 to 3 direction via AT and GC base pairing. So that's how the message is relayed. Why leading and lagging? Well, notice the leading is continuous. So imagine, to use a crude analogy, this is a zip on a jacket. Then we open up the zipper and we've got great access now. We can just go zoom, 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 and we can start to make this strand of DNA unimpeded. As the zip opens, I just make more. The lagging strand is fragmented. Notice, because I'm making exclusively in the 5 to 3 direction, because the old strand in the old lagging strand is actually already in the 5 to 3 direction here, because the old strand here must be 5, right? Because the other old strand was 3. So this old strand must be 5. Well, I've got to read in the 3 to 5 direction, so I can't just read that way. I have to go in to the congested part here, and then I've got to synthesize in that direction. So you'll notice that I'm synthesizing in, at least in this diagram, in the right to left direction for my leading strand, and I'm synthesizing in the left to right direction for my lagging strand. So I'm synthesizing in opposite directions. And I have, as a consequence, I have fragments that have to be sewn together. Fragments because of congestion. These fragments in the lagging strand are called Okazaki fragments. And it lags behind. It literally takes longer to make this. Why not just have one way to make it? Well, if the goal is to relay the message faithfully, you might argue that statistically that's more likely if you have one mechanism. If you were to read both in the 3 to 5 and the 5 to 3 direction, you'd need 2 mechanisms. If you only read in the 3 to 5 direction, you only need one mechanism. And the fact that this is what we see, this is what won, so we can assume that the other one was less faithful and lost the battle for evolutionary supremacy. Okay, so leading and lagging strands. Okay, what do we have here? This slide just essentially says the same thing. Again, how do we know? How do we know what strand of DNA to lay next? It's governed via these complementary base pairings. This is probably a great, I don't think I can get this video done in an hour, so we'll probably quit here and I'll make a short second video. But essentially what we've seen is probably this slide is a good place to just conclude what we've seen. So we've seen that DNA is the message in the bottle through time. The ultimate goal of DNA is to make protein, and then protein makes everything else. Enzymes and what have you. Fibrous tissue, globular proteins for different types of catalytic process. But the DNA also has to make copies of itself. So the DNA is a message in the bottle to make more messages in the bottle. And then it also has a secondary pathway to make the actual goal of the message, which is protein. So that's what we achieved in this video. And that will conclude this video, and this video is one of 2 videos.